Hi guys, welcome back to lecture 2 in classical genetics. Remember from the previous lecture that Mendel had carried out a number of experiments and he got some interesting data that showed this 3-to-1 ratio in his F2 generation, and this allowed him to formulate a mathematical model to explain what he saw. It also allowed him to formulate a couple laws. The first law Mendel wrote was called what we call random segregation. And what random segregation refers to is Mendel's conclusion that each individual has two copies of, uh, of each gene, one from his mother and one from his father or her father. And when organisms, sexually reproducing organisms, make gametes, those gametes are unique in that they only have one copy of each gene. And when the pollen gets together with the ovum or the sperm meets the egg, now you get a new organism with two copies of every gene. Okay, so random segregation refers specifically to the fact that during gamete formation, genes are split. So you have two copies. When you make sperm or eggs, each of those eggs or sperm only gets one copy of the gene. That's what random segregation is. And it's random because it's in a 50-50 proportion. Half your sperm would have the eye color gene your mom gave, and the other half would have the one your dad gave. Mendel's second law is called independent assortment. And what Mendel noticed, and this is a place where you can look at Mendel's data and see that he cherry-picked some of his data to support his conclusions. And what he said was that all these characteristics are independent of one another. So what makes sense is that a purple flowered plant shouldn't necessarily have a yellow pod or a green pod. That makes sense. Mendel asserted that all of these factors of inheritance, all these genes were unconnected, but that's not true. And of course, we know that because genes are carried on chromosomes. And we'll get to that later. But Mendel's law of independent assortment specifically speaks to the fact that different traits shouldn't be connected. So remember, we've talked about a number of different terms. A character or characteristic is a specific observable quality that you show. Traits are variants of that characteristic. So the characteristic would be eye color. The variants or the traits would be blue color, eye color, or brown eye color. Heredity is the passing of traits from parents to offspring. Remember that Mendel identified dominant and recessive traits. And that last but not least vocab term, allele, remember that alleles are just different copies of the same gene. When we talk about the genes that you have, there's a couple new terms that are introduced. So, for example, for eye color, let's just assume that brown eye color is dominant to blue eye color. And so we'll use a capital B to represent brown eye color and a lowercase b to represent blue eye color. And we'll call them dominant recessive. If you are homozygous, you have two copies of the same gene. So you could have big B, in which case we'd say you're homozygous dominant, or little b, and in that case you'd be homozygous recessive. Of course, homozygous means you have two of the same. Heterozygous genotype is big B, little b. And so in this case, the organism would have two different alleles, one copy of each. So, genotype. Genotype's easy. It's the type of genes you have. Are you big B, little b, etc.? Your phenotype are the traits that you express. So that's what you look like. Do you have brown eyes or do you have blue eyes? In flowers, we can talk about genotype and phenotype simply using this model. Let's assume red is dominant to yellow, so capital R represents red, lowercase r represents yellow, and we have three possible genotypes, big R, little r, or little r. That only gives us two phenotypes, though. You're going to be either red or yellow. And remember that the homozygous dominant, big R, is going to be red. The homozygous recessive, little r, is going to be yellow, but the heterozygote, big R, little r, will show that dominant trait and be red. Therefore, there are only two phenotypes, three genotypes, two phenotypes. when we look at these traits, we use the Punnett Square. And one of the simplest forms is the Monohybrid Cross. In this case, note that I've taken my parental gametic contribution, the gametes that they have, and I've split them up to make this box. And so in this case, what you see is a cross between two heterozygotes. Two heterozygotes. You can see at the top of the box and on the side of the box, those are the parental contributions. And up top, dad can give either big B sperm or little b sperm. Mom on the other side makes big B eggs or little b eggs in roughly equal proportions. And so we can model what will happen, or we can model what we predict to happen. So in this case, note that there's three possible genotypes: big B, big B, big B, little b, or little b, and they will show only those two phenotypes in roughly the 3-to-1 proportion. So this was Mendel's model to explain his results in the F2, and that brings us to the idea of probabilities. You could say in this case that the probability of having blue eyes, little b, is 1 in 4, or 25%, or 0.25. And of course, probabilities an important part of understanding genetics. If you flip a coin, you have a 1/2 chance of getting heads, or a 1/2 chance of getting tails. If you flip a coin 2 times in a row, the question becomes, what are the odds of getting 2 heads in a row? And the answer is that since those two events are connected, in the first trial, you have a 1/2 chance of flipping a coin heads, and then the second trial, but these are combined. So I have to combine these two terms, and I do it by multiplying. So the odds of getting 2 heads in a row would be a quarter. Of course, the same is true for having a boy or a girl. It's either one or another, and so the odds of having 2 boys in a row would be 1/4 as well. And we can see that using the Punnett Square. Now remember, this doesn't tell you what's going to happen. If I have 4 kids, that doesn't mean I'm going to have 1 blue-eyed kid. It's just a 1 in 4 probability each time of having that blue-eyed kid. So that's important. Here's some other simple examples, and you're going to get a chance to do a lot of probabilities in your practice sets. Remember terminology here. The parental generation is noted P, or sometimes P sub 0. The first filial generation is the F1. Second filial is the F2, so it's a product of an F1 cross. And here's his experiment. Here's Mendel's experiment with pea color. He had yellow peas and green peas. Yellow is dominant to green. In his first pea generation, he had a yellow plant with a true breeding green plant, and when he bred them together, they were all heterozygous. So look, homozygous dominant, homozygous recessive gives you all heterozygotes. And their genotype is heterozygous, but their phenotype is yellow. And so whenever you do your Punnett squares, you're going to talk about the genotypes and phenotypes exhibited and their ratios. In Mendel's F1 self-cross, or the monohybrid cross, he takes 2 heterozygotes, that is the offspring of this F1. They're yellow peas, but they're heterozygous, and he breeds 2 heterozygotes to get this classic 3-to-1 ratio here. So when this pollen met this ova, we get the homozygous dominant, etc. And what we see here again is that there are 1, 2, 3 different genotypes, but again, only 2 phenotypes, yellow versus green. And we can express them as ratios. So a quarter of the offspring should be big Y, big Y. A quarter of the offspring should be little y, and then 2 quarters, or 1/2 of the offspring, should have the heterozygous genotype. Now, in terms of phenotype, we've got yellow, 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 75% yellow, and 25% green. So that's it for the Mendel's model for the random segregation of alleles. And in the next cross, we're going to see a way to talk about his second law of random segregation. So I can look at one trait like eye color or P color, or I can look at two traits at the same time. And that's what a dihybrid cross is. So let's assume we have a plant that has round peas that are yellow. And we'll call it big R, big Y, little r. Remember that in Mendel's law of random segregation, if this is your genotype for these two traits, then when you make sperm or pollen, each of the pollen will have only one copy of each gene. So one possible pollen gamete would have a big R and a big Y. Another possibility is a big R and a little y. Little y, or big Y and little r, little y. So that's an illustration of the first law. Remember, his second law is this, that it doesn't matter what your pod shape is or pod color, they'll just simply randomly assort. And again, remember that that's not always true. I mean, it makes sense that maybe purple plants could have green or yellow pods, but doesn't it sort of strike you as odd that you could have a yellow pod with green peas in it? Anyway, typically in a dihybrid cross, you'll take organisms that are heterozygous at both loci, that's the location. So I've got two plants here that have round peas that are yellow, but they're both, their genotype is heterozygous at both loci. So both parents have the possibility of making these four possible types of gametes, and then we put it into the big box, all right? So here are the four possible gametic contributions of the father, and here are the four of the mother. And when you fill in the boxes, you get this stuff, which isn't a mess. It's kind of fun to figure these things out. When I cross this pollen with this ova, I get this new offspring, or that seed. And they will be homozygous dominant at both locations. Of course, they'll be round and yellow. In fact, so will they, so will they, so will they. Round yellow, round yellow, round yellow. Here, also round yellow, also round yellow. Another possibility is this round green phenotype. Round green, round green, round green, or wrinkled in yellow, and then finally wrinkled in green. And what you see in the dihybrid cross, where two heterozygotes are bred together, is this classic 9:3 to 3:1 ratio. So when you're taking the test and you see the dihybrid cross, be looking for this. What's the likelihood of having the wrinkled green or the homozygous recessive at both loci? Well, it's 1 out of 16. What are the odds of getting the dominant phenotype at both loci? Well, it's 9 of 16. There's 16 possible outcomes here. Last but not least is the test cross. If I have blue eyes, you know my genotype, right? It's little b, little b. But if I have brown eyes, do you know my genotype? Well, I know your phenotype, it's brown, but I don't know your genotype. Because brown could be big B, or it could be big B, little b. Well, a test cross is when you take somebody with brown eyes and you breed them with somebody that has blue eyes. If they have any kids with blue eyes, then they were heterozygous. Well, we spent a lot of time talking about this dominant recessive mode of inheritance, and guess what? It's kind of a rare way to inherit traits. What do you mean? I'm like, well, you don't really have blue eyes or brown eyes, do you? You're not tall or short. You're not smart or dumb. And so the truth is that characteristics inherited in this dominant recessive mode are relatively rare. Remember, in humans, in our class, we said that maybe tongue rolling is one of those dominant recessive traits. You can either do it or you can't. So what are the other ways that we can inherit traits? That's what we're going to talk about next.